From Microbe TV, this is TWiP, This Week in Parasitism, episode 173, recorded on July 12th, 2019. I'm Vincent Racaniello, and joining me today here in New York City, Dickson, Despommier.
Hello, Vincent.
And from a remote location, Daniel Griffin.
Hello, everybody.
Hello, Daniel.
It's been a while since we recorded, hasn't it?
The triumvirate has
like a month.
Yeah, it has been. That's true.
That's because you've been off fishing.
I was.
Daniel, were you somewhere?
I'm working.
Good.
I feel like I've been working straight.
Some of us have to work.
You're accumulating equity. Just look at it this way.
Well, I'm seeing fascinating stuff. So.
Oh, great.
We have a letter here from Folka, who writes, Dear Twix listeners and fans, this is a letter to you. There must be many of us enjoying the science and banter that Vincent and the team bring to us at least once a week. But if you look at Patreon, only 200 of us have already decided to say thank you by giving a small amount each month. Are you one of those listeners who really like Twix? Are you fortunate enough that you can easily afford a cup of coffee? Maybe you already considered to become a Patreon. Then please press pause right now and do it. It's really easy. It will not reduce the number of coffees you drink. It will make a real difference to the Twix team. Just imagine if Vincent and the others would only consider to do the podcasts, but not actually do it. Thank you. Greetings from climate change hot Germany, Folka. Thank you, Folka.
Indeed.
Daniel, what is our case from the last TWiP?
Great question.
I had paused and I was donating to us, but no, let me.
No, you don't have to donate to us. You're us.
All right. Let me remind everyone who is tuning back in and acquaint everyone who's tuning in for the first time with the case from TWiP 172. We discussed the case of a retired U.S. Navy physician who lives aboard his sailboat, travels about the world, and had just finished doing remote medical volunteer work in Central America. He's now back up in the Dominican Republic. He's in port there when he has the sudden onset of this dull and then progressing, worsening epigastric. So this is abdominal pain. So he's got abdominal pain. Over the next 12 hours after the onset, it really intensifies to the point where he's near tears, and he goes to a local medical center. He's admitted. He's given IV fluids. He's given probiotics. They note that he's got a fever. He's got an elevated white blood cell count. His platelets are low at 20,000. There's a left shift that I think we've talked about before to his white blood cell counts. He describes as far as symptoms that initially associated with this, he had some constipation followed by diarrhea, and there was some blood in the diarrhea. They discharged him without any diagnosis. He's still having fevers and chills. He then develops a rash with rose-colored spots on the torso. Right about here is when I get involved. And the initial recommendation was to try a 3-day course of azithromycin. Symptoms continue. And then at this point, he's started on metronidazole. Following the start of the metronidazole, he starts to feel better. The fever, the pain, everything resolves. And a few other things we asked about. Anyone else sick? Well, his wife was okay. But then it turned out that there were 3 men that were working on his boat. One of them had bloody diarrhea prior to when the patient did. And then there was a story of a bowl of nuts that apparently that man was eating out of. This man was eating out of. And we left everyone to try to connect the dots.
Mm-hmm. Those red dots.
Yes.
Dickson, would you be so kind?
I would be pleased to be first. Vic writes, Dear all, first of all, I would like to hazard a guess about this week's case. Due to the hint provided by Dr. Griffin as to the effect of treatment with metronidazole, and a quick browse of the 6th edition of Parasitic Diseases, I believe the retired doctor was suffering from amoebic dysentery. caused by Entamoeba histolytica. Gotta watch out for those peanuts laced with fecal matter. Secondly, I have a quick cruise ship parasite story. I work as a port agent here in Kristiansand, Norway. One of my primary responsibilities is to arrange medical treatment for sick passengers and crew members. So, over the years, I have seen most of the viruses and microbes that commonly plague cruise ship passengers and crew members. However, up to last week, I hadn't had to deal with a parasite. Last week, I received a request from the medical center on board and an incoming ship to take a crew member to the doctor and fill his prescription for permethrin cream and shampoo. Permethrin cream and shampoo. I went to the Feldskadlangens, which is an online resource where anyone can check the availability, price, and usage for all medications sold in Norway, and searched to see if I could get permethrin. I saw that it was available, and then I read the prescription of the usage, which was a bit unsettling. I contacted the ship to ask about the crew member, and I was about to go pick up and transport it in my car. They told me he was a newly arrived South Asian man who appeared to have scabies on his hands when he was examined by the medical center. To say I was a bit skeptical about hauling him around would be an understatement. Also, I heard from the staff that he was in isolation on board because they were very concerned about a scabies outbreak among the crew. After some back and forth, it was determined that the best course of action was not to go to the doctor, but keep the guy isolated and provide him with the cream and shampoo so he could treat himself in the cabin. I assumed that there was also a lot of scrubbing, vacuuming, and disinfecting that went on along with the treatment. I was not disappointed to hear that I wouldn't be doing the same scrubbing, vacuuming, and disinfecting in my car. Love the podcasts. Keep up the great work. Best, Vic.
Daniel, would that be an issue?
Which part?
Just having
Right, exactly.
So yes. It sounds like they're describing a crusted scabies, which is a severe form of scabies, which is highly transmissible. And I'm not going to use the Norwegian terminology.
You have to in this case. Come on, the guy was from Norway.
What's the Norwegian terminology?
It's called Norwegian scabies, unfortunately. But it's usually for immunosuppressed patients, though. Isn't that correct?
So you say that, and in the United States, yeah. Well, no, in the United States, we often see that, and there was a lot of it early in the HIV/AIDS epidemic.
Exactly.
But when you go to the tropics, you go to a lot of these places. In the Dominican Republic, for instance, it was so common to have such significant scabies that the people with scabies on their hands were important participants in some of the religious ceremonies that required these incredibly roughened hands. So when you go,
really?
Yeah, so when you go to the tropics, a lot of these areas, some of the villages that I go to, 10-20% of the population has scabies, and a certain percent of them will have this crusted severe scabies. So I think that's what we're seeing here. We're seeing a man probably from an endemic area, Southeast Asia, probably from an area where there's a lot of scabies. So he has one of these super infections, which, yes, historically it was actually described as Norwegian scabies, what we now call crusted scabies. I think of Norway as a low-risk place in general for scabies. Many others. But yeah, the crusted severe scabies is very very transmissible. I think we have some pictures in our book of.
I'm about to look it up.
Yeah, if you look at the hands, there's a photo of someone with crusted scabies on their hands.
Correct. And Bob Gwads, who used to give this lecture here at Columbia's Medical School, used to always refer to it as Norwegian scabies. And the reason for that was that there's also a reference to a Polish infection with head lice, and so he wanted to get even, I think. I'm not kidding. It's in the 6th edition. It's on page 498. And it said, figure 39.11, patient with Norwegian scabies showing numerous lesions, millions of mites may be present. So, I mean, it's a really disfiguring manifestation of an otherwise kind of benign, subcutaneous to cutaneous infection that usually
it makes you itch a lot, and
it does.
And the poor kids. I mean, this is, he's scratching himself. He won't stop scratching at night. And,
Right.
It could be,
They say he was newly arrived from South Asia, so would it be more common there than in Norway?
Ask the doctor.
Yes. Certainly. And certain areas in the tropics, as mentioned, really high rates. And then when people are able to come in and provide therapy, and someone who has it as severe as this, often in addition to topical therapy, because it's pretty hard to cover the entire body with the permethrin or the scabicides, we'll actually use good old ivermectin.
Yes.
So an adult, probably 5 or 6 of the tablets. A younger kid, you're going to, drop it down based on weight. And then often we'll repeat that 2 weeks later. But you do want to try to not let everyone else get the scabies.
And there's no immunity, so you can catch it again, unfortunately. That's the tragedy of this thing is it's in the superficial layer of the skin, so it really doesn't encounter the immune system.
Yes, and so when I go to these places, how do I not get scabies? Right?
Yeah, how did you not get scabies? Or did you? Maybe you did.
You're seeing these people, and so I always demonstrate for the parents how to put the permethrin cream on the children, and I make a point of getting quite a bit on my own hands, which is sort of self-treating, because it's a risk, and certainly people I've worked with in these areas have. Ended up with scabies, so to speak. No, not so. They've just ended up with scabies.
It's interesting because those people develop rashes not where the scabies mites are. It's in various parts of the body, but it's not actually where the mites are found, which is usually in between the fingers.
Yeah, I make a point, I think, in some of my lectures in the tropics that the tropical presentation of scabies is quite different than the temperate presentation of scabies. And one of the nice things I always try to introduce is something called dermoscopy, where you can actually look at the skin and you can actually see the mites.
Yeah.
And then you could really confirm the diagnosis right then. Here I'm seeing the mites, and you go ahead and treat.
So address the concern that the scaly droppings from, let's say, a person that's hyperinfected would actually result in, if you contacted that material, that you would actually catch scabies.
Well, it's not the droppings, right? It's not the dry skin. It's the actual scabies moving from them to you. And these people can have a lot of scabies.
No, you're right. And shaking their hand is probably the way you would acquire this. So scrubbing down a ship from top to stern would
stem
not
to stern.
That's from port to whatever. Come on, you're the sailor. The point is that has absolutely no bearing whatsoever on the way this thing is transmitted. So that wouldn't do any good anyway.
It's really going to be directly from them. So I understand the
concern, but not the approach. That's right.
Yeah.
Daniel, can you take the next one?
Sue Ellen writes, first off, I want to thank all 3 of you for the animated and informative discussion of my email about toxoplasmosis and indoor riding arenas in TWiP 172. My hypochondria meter is back down to 0, and I'm no longer worried about inhaling toxo in the indoor arena. Now for the case study. This was a tough one for me. I came up with 2 possible diagnoses based on the symptoms and probable location where our Navy doctor acquired the parasite, Central America and the Caribbean. Balantidium coli and Entamoeba histolytica, but neither of these will produce a rash on the abdomen. Back I went to Parasitic Diseases 7th edition. Strongyloides can cause a rash, but usually is asymptomatic. And our patient clearly had severe abdominal pain, plus diarrhea and bloody stools. So I'm a little stuck on this diagnosis, frankly, due to that rash. Could it be unrelated to the parasitic infections? Perhaps a reaction? Could he have both strongyloides and B. coli or Entamoeba histolytica? If so, my heart goes out to him. But I have to discount strongyloides since metronidazole is not listed as a possible treatment option. So I'm going with B. coli, even though the rash still puzzles me. I look forward to hearing what the actual diagnosis is. Cheers, Sue Ellen.
Great.
Marcus writes, Dearest doctors, sorry if this got a bit long. Hope I'm not too late with my guess. I've been finally catching up on my TWiP backlog after finishing exam season. I've been looking forward to this for months now. Though it may be considered a bit dull, my first thought when hearing stomach pain, sailboat, flagell, and parasite was an oldie but goodie, Giardia lamblia. This, however, only rarely causes dysentery. This makes amoebiasis seem more likely. Then again, Giardia might cause rashes. Don't think entamoeba does. Normally asymptomatic, amoebiasis is usually caused by Entamoeba histolytica or dispar and transmitted by the fecal-oral route. The risk of this is particularly high in cramped conditions with less than ideal hygiene and shared uncooked food. Peel it, cook it, or leave it. Sexual transmission by same route increasingly common. Normally causes abdominal pain and diarrhea with some blood and mucus. In some cases, more bleeding and fever, amoebic dysentery. The bleed would likely cause the light thrombocytopenia, and with infection, the left shift. Normally, the amoeba stays in the wall of the colon, but can eat way through to larger vessels and via the portal system to the liver, where additional damage is caused. Can, in rare cases, spread further. Diagnosis, ideally antigen detection or PCR, alternatively microscopy. Both bugs treated initially with flagell, followed by humatin or furamide to rid patient of remaining protozoa. Kind of wish it was giardiasis, since it would allow me to wax poetic on my favorite diagnostic test, the string test. But I'll have to land on amoebiasis. Either way, the treatment should work. Still loving the podcast from Levanger Hospital, Norway.
Oh, nice.
A second listener from Norway today.
What are the odds?
Wow. How about that?
Yep.
Dickson.
I'll take the next one. This is such
a long one, Dickson.
It is, but I'll try my best.
Sorry.
Tonto writes, Clostridium C. difficile. Next.
So you could take the next one.
Okay.
Dickson.
Thank you. Courtney writes, Dear Professors, hello once again from Omaha, Nebraska. I'm writing you for 3 reasons. First of which, the medical history podcast Sawbones recently had an episode about trichinosis. Well, that's interesting. Secondly, and I'll check that out. Secondly, in the previous episode of TWiP 172, Dr. Dickson stated near the end that there are no parasitic bees, but au contraire, there are indeed. Kleptoparasitic bees are fairly common, and they function pretty much like cuckoo birds. These bees are sometimes called cuckoo bees. They go into the nests of other bees, usually closely related species, and lay their eggs. Many times, these imposter eggs will hatch before the host, kill the other eggs, and then eat all of the resources. Finally, my guess. I have to admit, when I heard gastrointestinal pain and diarrhea, my mind immediately went to cholera. Reason being, I was currently reading Pandemic Sonia Sa, a non-fiction book discussing pandemics with cholera being the central disease. But of course, this is not cholera. And I might add, parenthetically, cholera does not result in bloody diarrhea. So I Googled metronidazole plus parasites, and that led me to this NIH article. He gives a reference. There, it mentions Giardia and Dientamoeba fragilis. While they do have some matching symptoms, there seems to be a few things that don't quite fit, such as the rash and the constipation. So I began looking at tables provided in parasitic diseases, finding a disease that was common among fever, diarrhea, and gastritis. Trichinosis is one of the diseases mentioned, but it doesn't mention anything about constipation or a rash. But looking at the table again, I see trichinosis is associated with leukocytosis. As mentioned in a previous email, I am not a medical person, more like an ecologist. Looking up leukocytosis on Wikipedia, I started to see that it does fit with elevated white blood cells and it being shifted left. But it's not treated with metronidazole or azithromycin, so back to the drawing board. At this point, I wasn't finding anything that matched up nicely with the symptoms. I had considered babesia and amoebiasis. Then I started to look at what azithromycin treats in regards to parasites. Turns out, not a whole lot, and even fewer diseases that match our symptoms. With this in mind, our patient has 2 infections. I think he has babesia, which explains the fevers and chills, and Dientamoeba fragilis, which would explain the diarrhea and gastrointestinal pain. I chose Dientamoeba fragilis instead of Giardia or trichinosis because these diseases are not related, or at least per Parasitic Diseases 7th edition, are treated with either azithromycin or metronidazole. All of the symptoms weren't listed, rash and constipation, but they could be red herrings. Thanks for the tricky patient.
Tricky patient.
I know. Good play on
like
words.
It's nice.
It's a good play.
Patient.
Good play on words.
Daniel.
Glenn writes, and this is a long one.
Indeed it is.
I wondered if the patient had amoebic dysentery, TWiP 17, in light of the bloody diarrhea and questionable response to metronidazole. He could have acquired it during his travels with a 2 to 3 week incubation period, or from the other person with bloody diarrhea on the boat. On the other hand, there are some additional clues that raise the possibility of typhoid fever. I know it is not a eukaryotic parasite. The rose spots on the abdomen is a classic, but not that common sign of typhoid fever. The patient was initially given azithromycin. Although this is active against Salmonella typhi, patients may take several days to respond, so the absence of a prompt response does not rule it out. Nuts may serve as a vector for Salmonella infections, and then he gives a reference there. And I suppose that in addition to acquiring an organism during his travels in Central America, the bowl of nuts could well have become contaminated by a carrier. Honesty alert. I am an infectious disease specialist at a public hospital that cares for a large immigrant population. Although we are not in the tropics, we certainly think of these conditions in our patients who travel from these regions. I recently started listening to your podcasts, including TWiP and TWiP, and found them an outstanding resource. You really capture the fun and excitement of ID that made many of us choose the field in the first place. By the way, we have found the BioFire film array PCR-based diarrhea panel a terrific diagnostic tool. Okay. Within an hour, it checks for 22 different pathogens, including Salmonella species and 4 of the common intestinal parasites, E. histolytica, Giardia, Cryptosporidium, Cyclospore. From my point of view, it has revolutionized diarrhea diagnosis and clearly improved our diagnostic acumen. Whether I am right or wrong, I always say that medicine is a humbling experience, so that my errors are all too common. I very much appreciate your podcast and will recommend them to our ID fellows. Keep up the good work, and thanks again. Glenn Matheson, MD, Department of Medicine, Olive View, UCLA Medical Center.
Very cool.
Nice. Right?
You bet.
We're gonna come back to this one, Glenn, so we'll have some comments.
Oh, yeah.
Kendra writes, hello, TWiP professors. It's currently a sunny 71F with 21C in Casper, Wyoming. Were you near there, Dickson?
Wasn't near Casper, but I was in Wyoming, and it was certainly sunny in 21C.
Kendra, Dickson was just in Wyoming for 3 weeks.
That's true.
Fishing.
Southern Wyoming.
You missed them.
Yeah, well, it's a big state. Come on. It is a large state.
My guess for the TWiP 172 case study is Giardia lamblia. G. lamblia has a cyst stage, which enables it to survive in the environment for long periods of time. These cysts are found in contaminated food and water and are spread by the fecal-oral route. They may also spread by person-to-person contact. The cysts are ingested and undergo excistation, releasing trophozoites. Trophozoites are the pear-shaped, binucleated, flagellated protozoa that emerge in small intestine and live on the surface of villi. They will encyst in the small intestine, and these cysts will pass with feces. Treatments for giardiasis include metronidazole, nitroimidazoles, and tinidazole. Hydration is also very important. Giardiasis can be diagnosed by microscopic observation of trophozoites or cysts, ELISA, nucleic acid amplification test, or direct fluorescent antibody test. Differential includes irritable bowel syndrome, tropical sprue, inflammatory bowel disease, cryptosporidiosis, traveler's diarrhea, and lactose intolerance. Best regards, Kendra. References, PD6, NCBI giardiasis. All right. Dickson, you hit the jackpot.
I certainly did. Now, this contributor to TWiP is not just a trivial contributor every now and then. This man...
Hold this up to Daniel so he can see it in front of the camera now.
I will.
Before you read the letter, Daniel, the author of this letter sent us this tome.
A year's worth of TWiP.
Isn't it amazing?
He self-published all of his letters, Daniel.
Look in the back. He's got pictures. He's got illustrations that go throughout this. Well, it actually isn't that cover. He's got a wonderful picture of a botfly with a myiasis.
When I see you on Monday, Dickson, you got to bring that with you.
I'm going to do it. Well, this is the only copy so far, but I'm going to get one for each of us.
I think in the book, if I'm not mistaken, there is a link where he has this as a PDF somewhere.
Yeah, would you rather have a hard copy, Daniel, or an online copy?
I'm a big PDF guy.
Yeah, I know. That's why I asked.
I think he might have. And somewhere there's a link to this, Daniel. I will send it to you.
It's a charming accumulation.
Well, we should put this on the website as well.
Oh, absolutely.
Oh,
yeah, if he's willing to.
Agree.
Isn't that cool?
It's wonderful.
I mean, he's got all of his multiple letters here. And Daniel, he writes here in the letter, this booklet contains 1 year's worth of my letters to TWiP.
Right.
And I don't remember how I discovered it, but the TWiP clinical cases hooked me immediately, and I've submitted a response to every episode since May. It is now May 2019. I decided to produce an annual and one handy pamphlet.
That's great.
In summary, TWiP has become something of an outlet for my need to explain something in this wormy world. The TWiP professors have a very light touch and have an absolutely non-commercial commitment to science communication and education. They are a rare breed. Isn't that lovely?
That's gorgeous. Yeah, absolutely. And we never fail to read his letters because they're very interesting.
Lovely.
They're interesting letters.
So anyway, I will find this link, Daniel, but we thought you would like to see that. It's lovely, Kevin. Thank you so much.
So Kevin has spared us the need for long, drawn-out articles to describe because his emails they fill up a lot of room, and they also contribute to our general understanding of medical history, history in general, anthropology. They cover a broad range of subjects, including the one I'm about to read. Kevin writes, he entitles his essay, "A Basket of Deplorable Nuts, Mixed Nuts and Mixed Guts, or Occam's Penknife." Our case involves a retired man, let us assume over 60 years old, doing volunteer medical work in tropical low-resource nations. He lives on a sailboat, currently docked in the Dominican Republic, and has exposure to various crew/employees that come from the regions where he works, one of whom has bloody diarrhea. Clinical picture is sudden, intense abdominal pain, fevers and chills, leukocytosis with left shift, constipation followed by bloody diarrhea. Rose spots described on the trunk. Initially treated with azithromycin without significant improvement, followed by metronidazole with prompt resolution of symptoms. I've got a problem with the concept of pathognomonic, occasionally spelled whatever. One problem is that so-called classic signs are oft recited triads of symptoms in reality have fairly low predictive value. Even worse is the tendency for such rubrics to be active system one thinking, where the reflexive associative thought process highlights the most available memory and swamps slow contemplation. This is all a rewarming of Kattenmann's ideas described in his 2011 book, Thinking Fast and Slow. I'm very good at the latter. Terms such as rose spots may immediately activate the availability heuristic, resulting in activation of closed circuits in my reptilian brain. Rose spots equals typhoid and encourages premature closure. It is often more valuable to employ objective language to describe clinical findings, rather than using leading terms such as rose spots. This may encourage diagnostic openness. A cumbersome but more objective alternative might state, patient with 10 to 15 blanching pink papules on trunk, approximately 3 to 8 millimeters diameter, almost completely unrelated to the foreign and mentioned solely in order to prolong this pedantic excursus. I direct your attention to A terminal curiosity in my endnotes, where past examinations of medical metaphors have been extended, in this case focusing on flower, vegetable, and plant metaphors inspired by the evocative term rose spots. Speaking of pedantry, I occasionally tire of Occam's scholastic razor. For this case, I prefer him to be armed with a penknife, allowing the multiplication of causes. Our patient's age, location, constellation of symptoms, those rose spots again, dysentery, dietary habits, that dirty, dirty bowl of nuts, and resolution of illness with azithromycin, followed by metronidazole, all lead me to conclude that our sailor has typhoid/paratyphoid fever and amoebic dysentery, both diseases. Rose spots can be seen in several infections, but typhoid is suggested by his initial constipation, intense pain, and thrombocytopenia. Medscape turns a nice phrase in describing typhoid as having notorious gastrointestinal manifestations. Azithromycin is an effective second-line antibiotic for typhoid, though our patient did not receive a full course. The rapid resolution of his bloody diarrhea with metronidazole is suggestive of Entamoeba histolytica infection. Several references are noted that discuss a protective or facilitating effect of helminth and other infections on typhoid. Another reference hypothesizes that Salmonella enterica infection protects Entamoeba from oxidative stress. I will also attempt to support my dual diagnosis by invoking Osler's 1907 text, where he describes 100 fatal cases of amoebic dysentery, 4 of whom had typhoid. The patient is now presumed cured. Should have stool/urine culture to rule out chronic typhoid carriage. Stool microscopy, antigen tests, or NAT should be performed to ensure that it does not have persistent amoebic infection. His nut bowl should be discarded, and his employees and spouse tested and treated if necessary. A postscript directed to Professor Despommier. Rose spots have been described in trichinosis. See 1985 KK Poon reference below. Thanks to the glamorous positive TWiP lococci, TWiP. I'll read that faster. Positive TWiP for their relentless, non-commercial dissemination of non-fake news. Do you want me to read the endnotes?
Just a few interesting ones.
They're all interesting. I mean, but I love this guy's writing. I really do. Endnotes and terminal curiosity. I'll just read a couple of randomly selected ones. According to Medscape, typhoid has notorious gastrointestinal manifestations. These include diffuse abdominal pain and tenderness and, in some cases, fierce, colicky right upper quadrant pain. An excellent website for all things skin. Yes, that as a reference too. What are rose spots? He raises the question. Rose spots describe the rash that occurs in up to 30% of people infected with Salmonella typhi. Characteristically, rose spots are seen in untreated typhoid fever. Rose spots usually occur the 2nd to 4th week of illness. Groups of 5 to 15 pink blanching papules, little bumps, appear on the anterior trunk. The papules range in size from 2 to 8 millimeters. They are usually distributed between the level of the nipple and the umbilicus, but can also be found on proximal extremities and back. Each lasts 3 to 5 days. Okay.
I want to go down to the next set of terminal curiosities.
Oh.
And as an addition to my discussion of zoological medical metaphors in TWiP 165
right,
and culinary metaphors in TWiP 159, I offer the following list inspired by the term rose spots with its pleasing floral associations. And these are flower, plant, vegetable, and medical metaphors, which include heliotrope rash, framboise, crops, cotton wool spots, ash leaf spot, cauliflower ear, gin blossoms, miliary tuberculosis, etc., cherry angioma, vegetative state, bamboo spine, prune belly.
Orange.
Anyway, these are all wonderful medical metaphors that are based on flowers and plants and vegetables. Isn't that cool?
It's great. Prune belly. I mean, I got a lot of this during my education here, and it stuck.
Sarcoma botryoides.
Right. I mean, it forces you to look these things up, and then you see their origins, and it's quite amazing how these things evolved.
Yeah,
I think he's a treasure of medical history, as well as acumen to see through the fog of the subterfuge of multiple symptoms and to try to distill it down into what's really going on.
Mm-hmm. Daniel, can you take the next one?
Yeah, I was going to say, I worry you guys are like, having a favorite child. I like all the emailers. I think they all have great qualities.
Yeah, no, I don't. Please, I want to be inclusive.
All right, so Ben writes, dear hosts of the wonderful Acanthamoeba podcast.
That's fabulous.
Nice.
I hope case guest makes it in time. I've been very busy trying to finalize my studies in Germany, so this will be brief. I believe the retired U.S. Navy physician is infected with Entamoeba histolytica. Like Dickson hinted at, the list of potential parasites is narrowed down significantly by looking only at those that resolve with metronidazole. I feel E. histolytica was the best fit, although I would not be surprised if there was secondary infection too. 2 weeks ago, I visited the Berlin Medical Museum, and then there's a bunch of German stuff there I can't pronounce properly, so I may not embarrass myself. And it's an absolute must visit if you're ever in Berlin. Part of the exhibit includes some of Virchow's original pathology samples, slides and drawings, and there is a lecture theater that was partially ruined in the war where he announced his cell theory finding to the scientific community. And there's a picture attached. Another incredibly confronting part of the museum has whole organs diseased and normal for comparison. That actually sounds fascinating, I must say. One thing I found hard to come to terms with was that every single organ that they had there, they had either a syphilis or a miliary TB infected sample. I never before appreciated how nasty these diseases really could be and how much of a nightmare it must be for a physician's differential diagnosis. We've had our warmest day for the year in Hamburg, hitting 34°C. Oh, my. And today I leave for the slightly cooler climate of the Swiss Alps to attend the upcoming Malaria Gordon Research Conference.
Oh, nice.
Thank you for all your wonderful work. And to all the listeners out there, remember, if you ever feel alone, Demodex follicularum will be with you every step of the way. Jay.
Please explain that to the audience.
So that's actually a mite that lives usually in our eyebrows. It's actually quite common. Most people have
that's right.
it. So it's, when people want to be completely cured, I'm like, you know what? You live in a sea of
microbial life.
So, so yes, you are not alone.
You are never alone. That's exactly right. Wow.
We have one more from Anna. Dear TWiP Trio, the sailor in the DR seems to have a case of Entamoeba histolytica. My guess is based on the rapid response to treatment with Flagyl and the symptoms being more consistent with amoebic dysentery than with Giardia, which should be easy to recognize since it smells quite distinctive coming out both ends. Oh. A colleague of mine contracted E. histolytica traveling in a low-resource setting, and it was quite debilitating. Sadly, it took her a while to get diagnosed after she was back home in the U.S. It just goes to show how important it is to make infectious disease education accessible to all, as you are doing with your fabulous podcasts. By the way, I'm a long-time listener, but I'm about to move to New York City, so if you do decide to have a live episode 200 in honor of Dickson's birthday, I would make a point of being there. It would be great to thank you in person for your outstanding work.
I'll do my best to be there, too.
Nice. We will have a 200, and we will try and do it live here in New York City.
Yeah.
I just don't know where, but we'll figure that out. We have some time.
We do.
Because we're at 173.
That's right.
We have about 10 years, right, at the rate we're going?
No, about a year or two.
We'll do it soon.
So we'll find a venue, hopefully with a bar.
Exactly.
That would be something, wouldn't it?
Well, it would be great.
It would be in the evening, and then people could buy drinks while they listen.
We have to do this by my birthday, which is June 5th, which is,
No, the 200th is not happening on your birthday, dude.
Well, we have to speed this up a little bit.
The 200th will happen on one of your birthdays, okay? It may be...
It could.
If it's 2 years from now, it will be your 90th birthday, okay?
Yeah.
You don't get that, do you?
I do. By that time, I won't even know it, and we won't go there.
Obviously, we can't do it for your next birthday.
No.
Well, we have two more people that haven't had an opportunity to weigh in on this case. So,
we do? Where?
I think Vincent, you want to weigh in?
Oh, right. Well, I tell you, for me, the dysentery is the clincher because Dickson once told me that E. histolytica is the only parasite that causes dysentery. Right?
Dickson, you're making an expression. Now, do you want to respond?
No, it's not the only. It's the only common parasite that causes dysentery. That's true. It is. Giardia does not cause dysentery.
No, the Giardia does not.
And Strongyloides doesn't either.
So when you think of diarrhea, right?
Well, Strongyloides was also called Cochin China diarrhea, but it wasn't a dysentery. It was a diarrhea, right? The difference is that dysentery has blood in it. So you have to think about that. Now,
so when you think about dysentery, so E. histolytica, what else is
Ballontidium coli?
B. coli.
Occasionally. And it's a rare parasite. I mean, it's a huge ciliate that rarely infects people, but when it does, it has some severe effects.
So I'm going with E. histolytica.
I don't blame you.
What about you, Dickson?
Well, I'm going with the Kevin's double,
huh?
Yeah.
Double trouble.
Yeah, because of the fact that, as I recall, Salmonella is it a facultative anaerobe?
Ask Daniel. He should know.
Daniel?
I'm going up because I think it's not just Kevin, because we also had someone else who
we did. That's right.
Through Glenn. Glenn
That's
was
right.
Also writing. So why do you ask about the facultative anaerobe? Because you're thinking
I'm
maybe
thinking that metronidazole kills all anaerobes.
It is a facultative anaerobe.
That's what I thought. So it's possible because azithromycin wasn't given to completion. And immediately after the third day, then he switched to metronidazole. So you had a double hit against anaerobic organisms. Azithromycin is not a specific for anaerobes, but metronidazole is. And so to get rid of his symptoms with metronidazole doesn't necessarily mean that it had to be Entamoeba histolytica causing the problem. It could have still been Salmonella typhi because it's a very effective drug against anaerobes. So I go with the double hit.
I don't think Daniel's ever had a double.
Well, that's until now, because the rose spots don't occur in Entamoeba histolytica. They just don't. So you've got these two separate things going on at the same time, and I think Osler's book of 1907 tells you that of the 100 patients that died from Salmonella typhi, only 4 of them actually had it. They all had something else. So you can't really assume that because you've seen 4 in a row and it turned out to be that the next 100 are going to be the same. And that's the problem with medicine. You've got to do the test and you have to work through it. So I'm going to go out on a limb on this one. Not really, because I feel confident that Entamoeba was there and Salmonella was there. And they're both transmitted the same way. Fecal contamination, oral fecal contamination. Mm-hmm.
Okay, so I will. I'm going to put this into context, and then this is going to be a good discussion, I think. So the way this case evolved was I got the call, and I was actually in Paris at the time. So we did this via a Skype call. We had to get the retired Navy person to an area where he had internet, and we talked. And I initially had the impression that there's 2 things that I'm worried about here. I'm worried about Salmonella typhi.
Right.
So enteric fever or typhoid, and I'm also worried about amoebic dysentery.
Right.
And so here was our plan. We said, first off, I said, the Salmonella, you're going to be able, you can survive that for several days. But if you have Salmonella typhi that goes untreated, you're at risk of a perforation, you're at risk of sepsis, you're at risk of dying. He said, so we have 2 choices. One is we just give you both, and we see if you get better. Choice number 2, which we realized was not an option, was we try to actually get diagnostic testing done. That really was option number 1, but that was unavailable. So we didn't have any ability to actually confirm the diagnosis. So we were going to be approaching this with treatment.
Yeah.
Option number 2, I'm going to counting 2 twice here or whatever, but another option was we treat the Salmonella typhi first, and then see if he gets better. And then we reserve treatment for the amoebiasis second. So that's how we approached it. We did 3 days of the azithromycin, and then we were checking in each day. And at the end of the 3rd day was the report of I'm on the azithromycin for 3 days, and I have not noticed any improvement. So there's a couple things at this point. One was that, and I think some people discuss this, azithromycin, you may be treated, you may not see a response until day 5. So here was at day 3, we had our decision tree. Do we say we'll give it 2 more days, or do we start our treatment with metronidazole? So we decided at day 3, all right, I'm not confident that it's not the amoebic dysentery. The nice thing about azithromycin is an incredibly long half-life. So we've treated him for 3 days. It's still in the system when we start the metronidazole. So we start the metronidazole as described. He rapidly feels better, but now he's got these rose-colored spots. And he sent me a photograph of those. And it really, I have to say, this is type 1 thinking that I think Kevin
pathognomonic.
When you see these rose spots on the trunk developing, at the right time in the right clinical context, then I went back and I said, okay, a couple of things I want you to do. One is, let's finish that course of azithromycin. I know you're starting to feel better, but I don't feel okay with a truncated course of treatment for typhoid. I can't confidently rule that out. So he did go ahead and he did his, I believe we did 7 to 10 days of the azithromycin. So he restarted. We also went ahead and we treated not only with the metronidazole, but we also did a luminal agent. And that was quite something, finding out what we could get for that. So my impression was I could not rule out typhoid, Salmonella typhi. So we treated for that. And then we got another little bit of interesting history that helped. That man that we mentioned who was working on his boat, he actually was able to go and get a diagnosis. And what did he have in his stool?
Both.
Entamoeba histolytica.
Yeah, totally. He never does both, dude.
Yeah, so I went with both. I went with what Kevin, what Glenn, what Dickson. I said I cannot rule out Salmonella typhi and untreated. That is life threatening. So we did our course of azithromycin, but I also felt quite confident that we were also or treating the Entamoeba histolytica.
So what is the rose spots in case it wasn't Salmonella?
The rose spots really, they could be a couple things. One is they could always be a reaction to the medication, but I have to say a medication rash is usually diffuse. It does not look.
Right.
I have to say the rose-colored spots, when you looked at the ones that he sent me, it was right out of a textbook.
The other possibility is that the other person with symptoms was a carrier for Salmonella. So he wasn't suffering from that. He was actually suffering from Entamoeba histolytica, but was also a carrier for Salmonella, which would have still placed the captain of the boat at risk from both infections.
Yeah, and it is. I mean, this is your classic case in a limited resource setting where you've got to say, I can't just say, oh, this is pathognomonic, I'm just going to treat what I think it is. You've got to say, this person's going to die if I don't do the right thing here. And so, giving an individual 2 medications, for a week is not such a bad thing.
No, I think you did great, by the way, and I'm sure that the captain of the boat thought you did great also.
He was delighted.
Yeah,
and got better.
Must have been. And I'm sure he was more cautious the next time of his snacking habits.
Hopefully.
So, that's interesting. It's diagnosis by treatment. And I'm sorry to say this, but a lot of physicians are forced into that mode due to a lack of resources to actually get a definitive diagnosis.
Well, I think as Glenn points out, I love the BioFire. I love the ability to get a diagnosis much more often than historically we had been able to, particularly in gastrointestinal situations where so many times, you send off a stool culture, it never grows, you're treating empirically, you're never sure what you were treating. But now with the GI PCRs and a lot of this new technology, we're doing a better job of knowing what we're treating. And I think that feeds back into our ability to then recognize clinical syndromes, confirm what our impression might be, rather than, in a sense, sort of a confirmation bias, right? Most people get better no matter what you do, and then you think it's because you did the right thing. Now we more often have the diagnosis, and we can associate it with symptoms in our treatments. So,
yeah.
All right. Should we give away a book?
You betcha.
We had 8 people I identified as not having previously won a book, so we will get a random number between 1 and 8.
Start the drum roll.
Is that a drum roll?
That's a semi-drum roll.
Whatever that was.
That's the sound of one drum drumming.
It's number 4.
Right in the middle.
Courtney from Wyoming.
Cool.
All right, Courtney.
Cool.
You are the winner of PD6, right? Because we don't have 7s yet. Do we have some autographed PV6s here?
We do.
And just send me your address, TWiP@microbe.tv.
Yeah.
And you're in Wyoming, so that's all I need is your address.
Can I go hand deliver it, please?
Yeah, go. You were just there, so you can't.
No, I want to go back. It's a beautiful place. Yeah, it's gorgeous.
You don't want to do podcasts anymore?
No, I didn't say that. I could do it remotely. I can amend my behavior. I loved it. I mean, I had a great visit out there this time. It was just wonderful.
Hey, Daniel, you been to Wyoming?
I've spent a lot of time in Wyoming. My parents actually at one point were looking at retiring to Wyoming. I love Wyoming. And my daughter actually right now is in Montana, so just right north of there.
Wow. What part of Montana is she in?
She's staying in Missoula, but they're actually out backpacking
great.
at the moment, so out in the wild somewhere.
So Vincent and I shared some experiences in Missoula, and we had a great time.
Well, we just flew to Missoula, but we went to Hamilton.
Yeah, which is only 40 miles south.
It was fun.
Fine. Yeah,
we had a good time. We did a podcast.
We did.
A TWiP.
Yep.
You took me fishing.
I did.
Well, I sat and watched you.
No, you did the video. You did.
And there's video. Someday I'll release the video of you fishing, catching one after another.
I tell you.
And then we had a couple of nice dinners, right?
We did. Yes. And we shared
And I don't think we argued at all. Right?
None. We had some amber fluid that we shared.
It was very nice whiskey. Was it whiskey?
Yeah, it was single malt. Yeah.
We sat behind our hotel by a mosquito-infested lake.
Exactly.
Watched the sunset, but it was fine.
That was good.
It was lovely. Maybe we should do it again sometime.
I'd love to.
Wyoming is a nice state, right? I've never been.
Gorgeous.
What's the town, the main place?
The main place? Cheyenne.
Cheyenne.
Then there's Laramie.
I think Jackson is the biggest town, population-wise,
right now. Jackson, yeah.
I think it is.
Where did you fly to, Dickson?
I flew to Salt Lake City and then rented a car and then drove
okay.
hour and a half to this little town of Kemmerer, which no one has ever heard of except if I tell you that that's where the mother store for JCPenney was. Then, that Kemmerer. It's also the diamond capital of the United States. And the opal capital, except for it's not quite as good as Arkansas, but it's got a lot of natural opals there as well.
I'm checking populations, and I think Cheyenne beats out.
Yeah, I do too. I think that's the capital.
Yeah, so 63,000. When I was in Fort Collins, it was about an hour to Cheyenne, so Cheyenne was closer than Denver.
Yeah.
I like Fort Collins a lot.
Yeah.
In fact, ASV is returning to Fort Collins.
Oh, really?
Years, yeah.
Ooh, count me in.
You like Fort Collins?
Oh, well, I like that whole area. I mean, I just love the West. I fell in love with it. How's that?
Let's see. Next summer, Fort Collins.
Wow. I'm there.
June 13th,
Let's
2020.
do another live podcast.
Colorado State University. I always do a TWiP at ASV.
Let's do it.
But I could live in Fort Collins.
Oh, yeah.
I could put a little studio downtown, but no one would see me. So.
Isn't that where your office was, Daniel?
So I was actually the chief of medicine at the hospital in Fort Collins.
Exactly.
Daniel, why don't you go back and we can make a studio and do podcasts?
Oh, that would be great. Oh, I would do that.
Yeah, Daniel wants to stay here.
Well, not forever.
All right, we have a paper.
No, we have a heroine.
Well, the paper.
No, paper first.
The paper? I'm sorry. Okay, sorry.
We have a shtick here.
I thought I read that right.
Can't you just mute him there when he...
I need a switch. I need a mute switch for Dickson, right?
You do. You actually do.
All right, this is a bioarchive preprint, which was recommended by Ben. Ben writes us a lot. He wrote about the Berlin Medical Museum. Let me try it. Berliner Medizinhistorisches Museum.
Close.
And he said you should do this paper, which is a bioarchive, which means that it's open access, and it's probably been submitted somewhere. It's called Matryoshka RNA virus 1, a novel RNA virus associated with Plasmodium parasites in human malaria.
Amazing.
The authors are Sharon, who's the first author, and Edward C. Holmes, the last author, who I know. He is a well-known Virologist. He does bioinformatics of viral sequences. He's in Australia at the moment.
Mm-hmm.
I just noticed as we're looking, Nick Enstay is one of the authors here. And he's a guy that I was involved with doing some research on malaria in children in Tanzania many years back.
That's right. I think Ben pointed that out.
I'll be darned.
Yeah.
That you had said at one point you knew him on the podcast, I guess.
Yep.
All right. So there are some parasites, there are some viruses of the parasites we talk about on this show, but not a lot. And we've talked about a few of them on TWiP. TWiP 126, we talked about immunizing against the virus ameliorates exacerbated leishmaniasis.
Mm-hmm.
TWiP 47, for whom the trick tolls, exacerbation of trichomoniasis by a double-stranded RNA virus. That title, Dickson.
Yes.
Toll refers to toll-like receptors that detect
Indeed.
the RNA and make the trichomoniasis worse.
That's right.
Isn't that a wonderful reference? For whom the bell tolls, for whom the trick tolls. You get it? Ernest Hemingway.
I got it, Vincent.
So there are a number of viruses of parasites, but not a huge amount. It's limited. As they say, our knowledge of protozoan viruses is clearly limited. And why is this important? Because in those two TWiPs, we showed how these viruses can modulate a parasitic infection, make it worse. Now, in fact, there are no viruses that are known to infect Plasmodium species.
Until now.
Until now. And did you know there are 100 species of Plasmodium that infect mammals, birds, and reptiles?
I did. I actually have the definitive book on that on my shelf.
100.
It's written by Garnham.
So I know Plasmodium falciparum and Vivax and Ovale.
And Knowles Eye.
Yep. But what are some weird ones I wouldn't know?
Chabode.
Chabode?
Yeah.
How about Cynomolgi?
Cynomolgi. That's a fairly common one, actually. It's in primates.
Any others?
There are some mouse and rodent Plasmodia.
What's the one that they use in mice all the time? Is that Knowles Eye?
Blocking. No, that's primates. I'm blocking on it actually right now. It's
okay. I won't put you on the spot, Dickson.
It didn't put me on the spot.
Even though I already did.
My memory is failing.
Anyway, the point is, up until this paper, no viruses of Plasmodium species. And so that's what they set out to do here.
That's right.
The way they did it is they obtained the parasite from people. All right. They got 3 species, Vivax, Knowles Eye, and falciparum, from 7, 6, and 5 malaria patients in Malaysia. All right. Uncomplicated malaria. And let me see if they purified the parasites. Does anyone remember offhand? Or do they just do total blood? Let's look. Let's look at the methods. Let's go to the methods, Dickson.
It would be difficult to purify the parasites.
See, having an expert like you on the show.
They're inside red cells, and that's going to be tough to separate the infected from the non-infected red cells, I think.
So you think whole blood?
Yeah.
Let's look at the okay, so that's wonderful that you could come up with that. I want to see what they say here. So, okay, total RNA extracted from blood. Just take blood from these patients.
It requires an enormous kind of laboratory to separate the parasites out from the blood. So I think that's, given limited resources, do the whole thing.
Okay, so you take blood, you extract RNA, and you sequence the RNA.
And it's not just whole blood. I'm sure they eliminated the white cells first to make sure that it wasn't some viral infection in white cells, because that's not possible for malaria to infect.
Okay, that's fine.
So I don't know if you need to, because I'm looking at, I guess we should find the methods in this, because if you look at the figures, the way they're approaching this, one of the things you can do with the wonderful molecular technology is you could generate all these raw reads and you trim them down and do your sort of cleanup, and then you can actually remove human known sequence.
That's right.
And then you're left with what's left.
Sure.
So you can basically
That's true.
subtract stuff out.
That's right.
Yeah, in fact, so they subtracted the human ribosomal RNA, and that's huge because most of the RNA is ribosomal RNA.
Exactly.
And then they made contigs, these are short reads, these sequences, and then you can make bigger overlaps, make bigger. And then they say, let's then search databases. And what they did was there's a database with all RNA-dependent RNA polymerase protein sequences. And that's the most conserved protein among RNA viruses.
I'll be darned.
RNA-dependent RNA polymerase.
Wow.
And they found hits only in the Vivax library.
That's interesting too, right?
In the end, they pull out a virus with homology 42% identity to a Narna-like virus, which was identified in mosquitoes. And this was pretty abundant, about 1.6% of all the non-ribosomal RNA. And they also had a lot of contigs that were unknown. And they found one in particular that was highly expressed, and it looked a lot like it would be a second RNA of this virus. It has high similarity to the one viral RNA they identified, and so they think it's the second segment of this virus.
Which makes sense. I mean, as we're sort of going through, the way they're doing this is they're basically taking a huge database, and the fact that there's human lets us know there probably are white cells in here, right? Because there's no not going to find a lot of genetic material in our red cells. And then they've got all this data, and they're digging through and basically looking for matches, hits, so to speak. And since the reads in most of this technology are short, you're not getting the entire sequence. So you're looking at overlaps, you're piecing stuff together, and sometimes they're going to not quite overlap.
So, Dickson, we just got to the point where they've identified a Narna-like virus, plus-stranded RNA virus, and theirs, they think, has 2 RNA segments. And most of the Narna viruses we know of have only 1 RNA.
I'll be darned.
So this is unusual. We'll talk a little bit about Narna viruses in a moment. So one of the segments contains the RNA polymerase, and the other 2 overlapping reading frames with proteins of no known function.
Really?
And they validate the presence of these RNAs in the Vivax samples by PCR. They are not found in Nolzi or falciparum. They're not in uninfected patient samples. And they call it Matryoshka RNA virus 1 because they say it's a Russian doll composition, a virus infecting a parasite infecting an animal. To which I say BS, because bacteriophages infect bacteria that infect humans, and we don't call them Matryoshka viruses.
But you could.
But why would you do that?
Well, because they wanted to be cute.
Well, in science, we don't do cute.
Oh, yeah? Look at the names of all of these super genes that control entire cassettes of things like the hucks, the hedgehog, this.
We do cute.
We do?
But I would say that
we do cute in science.
This Matryoshka is not unique to this virus. Many viruses are Matryoshka viruses.
Well, then now you've got an article for a whole family of viruses.
The double-stranded RNA viruses of Leishmania and Trichomonas, they're a virus within a parasite within a human. So why does this one get a special?
Well, Trichomonas is not an intracellular parasite, so it's not inside.
It's in a human.
Well,
they say a protist that infects an animal.
This is an...
Yeah, but they don't name it that because it's intracellular. They say that it infects an animal.
Okay.
A virus of a protist of an animal.
Okay.
Anyway, I just wanted to register my displeasure.
Gentlemen. And I'll jump on board because, Leishmania, right?
Sure.
So...
that's right. That's right. You're right. And trypanosomes have their own RNA viruses as well. So.
Yeah, I mean, if anything, as they start, we don't know a lot about viruses that infect parasites, but I think what we're starting to realize is that knowledge gap doesn't reflect a gap in existence.
No, that's right.
When we look, we find stuff. And the way they looked, you basically can go on we'll call it a fishing trip for Dickson's benefit. And I like the way they did this is you basically take blood that has the parasites in it. And I like the way they have these Vivax falciparum. So you really have an internal control that this is in Vivax. This is, not in, the other. And then, but I think that you could approach a lot of the different parasites this way. I bet Trichinella actually has some viruses. I don't know if they've
Sure.
looked at that, but you approach it with the same technology, you get Dickson back up and running again.
Well, Dickson, if you still had a lab and you had Trichinella,
I'd be dangerous.
We could do this for you.
I would be dangerous.
With you. We could collaborate.
We could do that. We could.
Too bad your tricks all gone.
I know.
No more bags of tricks.
No, I actually miss it. I'll be honest.
You told me once you didn't miss it.
No. I miss the excitement of a new discovery. That's basically what I miss.
Let me tell you a little bit about Narna viruses.
Yeah, please.
So this is the family Narnaviridae.
Are they from Narnia?
That's what I thought about.
That's another cute. I don't know. How is
The Lion, the Witch, and the Wardrobe.
These
right.
are very interesting. No, they're not from Narnia. So the family is Narnaviridae. There are 2 genera. All right. The first is Narnavirus, of which this is a member.
Mm-hmm.
The known Narnaviruses infect fungi, for example. They're Saccharomyces and others. They are capsidless viruses. They do not encode a capsid. They exist in the cytoplasm, and that's where they stay. They have no extracellular phase.
They're naked and afraid.
They're naked. I didn't want to say that N word, but yes, they're naked. So they remain in the cytosol. They multiply and they spread as the cells divide. So Saccharomyces divide, and the viruses go to the 2 daughter cells. Okay, so that's the Narnaviruses. One RNA that encodes only an RNA polymerase that copies that RNA over and over again.
Amazing.
Right? It's pretty minimal.
Yes.
However, within the Narnaviridae, there's another family. There's another genus, called the mitovirus. And these are viruses in mitochondria, which we talked about on TWiP, and which Brad Hillman over at Rutgers works on. And we're going to go visit him this summer and talk to him more about
That's
those.
a Russian doll.
Because it's in a mitochondria, and it probably got there when bacteria went into cells years ago and became mitochondria. They brought their viruses.
Is this anything related to any of the phage RNA?
Well, the mitoviruses are related to phages, yes, because they came in with bacteria. So that's a little bit about Narnaviruses. They infect fungi, plants, and protists now. This guy's right.
Right.
And the most related virus to this matryoshka is Wilkie Narna-like virus 1, which was identified in mosquitoes.
Huh. Which, by the way, transmits malaria.
I heard that they do.
So maybe that's how it got into the parasite.
All right. Now, there's some others. They do some phylogenetic analysis here, and you can see some closely related Narnaviruses besides Wilkie are Leptomonas Seymouri Arna-like virus, Phytophthora infestans. These infect unicellular eukaryotes. Mm-hmm. They do some work to show that probably Plasmodium is the correct host rather than something else that was present, some other parasite or other microbes that might be present. P. vivax is the right host.
Mm-hmm. I just thought of another species, by the way, of malaria.
What's that?
Plasmodium lofuri.
Lofuri, right. So birds, right?
That's correct. And William Traeger at Rockefeller worked on that organism for about 30 years.
We did a great story about that on TWiP, but it takes too long to go into it now. But the origination of a retrovirus from P. lofuri infected birds.
Really? Great
story.
Really?
All right. So what about other hosts? They look at bird samples infected with leucocytozoon. These are Plasmodium of birds, or
Plasmodium-like.
They say apicomplex and
that's right.
belong to the same Hematozoa subclass.
Right.
Okay. Yeah. And so they had sequence data already existing in the databanks from various tissues of Australian magpies, pied currawongs, and ravens collected in New South Wales.
Sounds like Corbides.
So they blasted the sequence against this, and they pulled out a sequence like their Narnavirus from leucocytozoon infected birds.
That's quite amazing.
So a related virus in a distant apicomplexan, right?
Vincent, did you know that birds have nucleated red cells?
Yeah, when I was a first-year graduate student, I worked on chicken red blood cells. I was studying their nuclear pores.
There you go.
And that I know that because they were there. There were nuclei in them.
Interesting.
Yes. Why do they have them and we don't?
That's a very good question.
Daniel, do you know why?
I actually don't. That's a good question.
I don't know why we would have them.
No, I don't know.
All right. So then we have another virus infecting a parasite of birds, and they found it in 6 of 7 birds they checked.
Isn't it remarkable that in this very small sample of Malaysian blood of people, they
maybe
immediately
it's quite prevalent.
That's the point.
And no one's looked before, right?
That is the point.
So they say these Plasmodium and leukocytozoan viruses are a nucleid. And
right.
So they're bisegmented RNA viruses. First documented RNA virus in Plasmodium. The 2 RNAs, one encodes a polymerase, the second, 2 proteins of unknown function. These are capsid-less viruses that remain in the cytosol. And so their effect on Plasmodium is unknown. But now,
is it possible to cure?
I was just going to say, maybe we can cure them.
With a antisense RNA?
The poor Plasmodium, we're going to cure them of their viruses.
So
that's right.
How would you do that, Dickson? How would you cure a Plasmodium of RNA?
Well, you can culture some of those malaria. You can culture Plasmodium vivax. Yeah. And you can introduce an antisense RNA to the RNA virus and see if it disappears. And if that works, then you can have a pure culture of Plasmodium vivax that has no virus.
Like an siRNA.
Yeah, exactly. A silencing or
and maybe it would all be degraded so that it would not. Yeah,
could be.
You could do that. It would be interesting. Now, Dickson, what if you did that and all the Plasmodium died?
One other thing, you could actually attack the RNA polymerase because it's specific for the virus and not.
Yeah, how would you attack it?
Well, haven't they characterized this protein pretty well?
Yeah, what would you do? Make an antibody?
Or
isn't there a
molecule?
Yeah, isn't there a drug that actually interferes with the
Well, you'd have to identify such a drug, yeah.
Hasn't that been done for other viruses?
Well, but this is not very similar to other polymerases, remember? It's only 40-some% to these Narnia viruses.
Yeah, but what about
Narnia?
What about the active side of the polymerase?
You could do it, but it would be a lot of work.
Okay, well, probably not worth it.
Probably, I would do the siRNA experiment.
Yeah, that's RNA. I would pick that one as the result.
But I guess the big thing is to sort of decide clinically the relevance, like as we've seen in certain the Leishmania subspecies.
Yeah, sure.
And trichomonas.
And trichomonas. It looks like sometimes the viral infection is directly tied to pathogenicity.
That's correct.
Or as we saw with the Wolbachia in certain parasites, it's actually like in a symbiotic relationship.
Exactly.
So these viruses may not actually be making vivax sick, so to speak. Yeah. It may be either making them virulent. There may be a symbiotic relationship here. And a lot of this, they sort of, they mention, but we don't know yet. What is the biological significance of these viruses in P. vivax?
That's right.
And this is relevant, I think, because some of the recent stuff we're seeing now that, again, we have better diagnoses. We used to say, all those malaria deaths in sub-Saharan Africa are due to falciparum. But we're realizing, actually, no, a more significant number are due to vivax and, other species. So it matters.
And the other, I'm sorry. Continue.
I was going to say, so it may matter.
Right. So vivax has an unusual feature about its life cycle that may be related to this infection that it has, in that it produces something called an hypnozoite. It's a resting stage of the parasite once it gets into the host that matures at a rate which is not dependent upon how many organisms infect the liver, but rather it's on a timer. It looks as though it's on a timer. And that the hypnozoite then lies dormant, let's say, for a year. And then after all the blood stages have been cured and the immune system kicks in, and then it starts to wane, up comes another hypnozoite that decides to initiate a new infection. Actually, it's an old infection, but it's a delayed infection. How's that? And that can last up to 5 years in people. So those are the relapses that Plasmodium vivax is typically known for. So it's possible, because Vivax is the only one infected with this virus, there might be a connection between the ability to make a hypnozoite and the presence of this virus.
So they had 7 Vivax samples from 7 patients.
Right.
They were all positive for this virus.
Guess what?
So let's do a bigger study now.
Yes, a much bigger study.
Where are we going to find Vivax infected
It's all over the world.
Okay, we'll get hundreds.
We will.
And we'll see if it's in every one of them or in a subset. And if it's in a subset, if it correlates with disease. Are there different severities of Vivax disease?
Ask the doctor.
So certainly, and along these lines, if we want to suggest that it's related to the hypnozoite, what other subspecies of Plasmodium has a hypnozoite
I was going to go there.
Go there.
Plasmodium ovale, which is a variant. It's a variant of Plasmodium vivax, only found in West Africa and several other related places. It's not as widely distributed, in other words, as vivax is, but it does have an hypnozoite stage as well. So it's possible to look. They didn't look there because they only took blood from Malaysian infection. So I would love to know whether or not Plasmodium ovale, which they didn't test, has this virus as well.
Yeah, because if it did, then, and those were the,
those are the only 2.
Yeah,
exactly. I think it's fascinating speculation. Whether it's actually true or not, we don't know.
Yeah, so there's, this is, I think there's a lot of interesting questions this raises.
Mm-hmm.
Interesting investigations to go forward with.
No question. By the way, Plasmodium falciparum has a unique feature that none of the other species that infects humans has, at least, and that is it's got some genes that are derived from plants.
Which ones?
That's a good question. I'm not sure which ones, but I remember seeing a Gordon conference talk on this once, and they said, can you tell which mice are infected with Plasmodium falciparum? They've humanized these mice to allow the infection to occur. And they had mixed infections of various other kinds of malaria too. And the investigator had dipped each of these white mice into a plant dye of green color. And the green mice stood out among all the other white mice. Those were the ones that had Plasmodium falciparum. We colored them green because it now turns out that they have genes derived from plants. So it's a more recent parasite of humans than most of the other species of Plasmodium. That's my current level of knowledge. I don't go beyond that, unfortunately.
So what do you think? Will this be found in all Vivax or just in a subset?
Oh, that's a great question, because I would love to know the answer to it, of course. And they're obliged to do those studies now. And
it's interesting. Very good.
Take it forward.
Very nice. Now, they've picked this one up because it had homology to nornaviruses, but there are a lot of
Bunya. They mentioned Bunya in there also someplace, no?
I don't know why. Yeah, they did mention Bunya. Why did they say
Isn't that a segmented virus?
Single-stranded Bunya and nornavirus-like. Oh, they said more recently, single-stranded nornavirus-like and Bunya virus-like RNA viruses were identified in trypanosomes.
Oh, that's right.
Actually, trypanosomatid, leptomonas.
Right. That's a parasite of plants.
And Crithidia.
Yeah.
Crithidia.
There was a lot of early work on trypanosomes that used Crithidia as their model.
But now, where was my thought going before you interrupted me? What was I saying?
How would I know?
Oh, yeah, here we go. So there were lots of unknown sequences which didn't have homology to any virus. It's likely that there are some viral sequences in there. So there are probably others that they couldn't identify. That's what I had to say.
Okay.
And I don't know. You just have to wait until those get annotated or someone else discovers that it's a virus. Or
Exactly. And the other thing that you know for sure is that the mosquitoes that transmit these Plasmodium or Plasmodia, they can transmit all 4 species or 5 species, by the way. So the same species, like Anopheles dirus, which is one of the vectors in Asia, that mosquito can transmit any of those 5 species of malaria. So they're not probably giving the virus from the mosquito to the malaria.
No.
That's what I'm trying to say.
there's no extracellular phase of these viruses.
No, but the replication cycle of malaria in the mosquito is intimate.
Let's talk about this.
Yeah.
Because this RNA virus would have to be in every stage of the malaria parasite.
One would think.
Right?
Mm-hmm.
So in the mosquito, we have which stage?
We've got the
Sporozoites.
sexual stages.
Sporozoites are in there? They deliver sporozoites.
They suck up the gametocytes.
All right, so they have to have the virus in them. Or
They're in there, and then they mate, and they produce an ookinete, and the ookinete then crawls through the stomach wall of the mosquito and embeds itself in the membrane, and then results in the oocyst. And the it goes from a diploid organism to a haploid organism very rapidly once it gets inside the stomach wall. And that's where the sporozoites are produced. And they're all haploid organisms.
the bottom line is that every stage of the parasite development has to have the virus in it, has to have this virus RNA in it,
I have a book on this.
because otherwise it wouldn't be transmitted.
That's correct.
But again, we don't know if it's in every
We do not.
infection, if every parasite out there, every Vivax out there has this virus or not. That's an interesting question.
There's no need to correct me because I was correct. It's called the ookinete as a diploid organism. And then once it develops into the oocyst, it starts to produce haploid organisms.
Anything else, Daniel?
Yeah, I was just going to say, when it comes to the mosquito cycle, I pretty much need to look at the life cycle. So the fact that you can walk us through the sporogeny without a reference is...
Hey, that's why it's in the book, right?
That's why we wrote this damn thing.
Yes.
That's exactly right. And
I think that this was a great paper, and thank you, Ben, for suggesting it.
Absolutely. But again, like one of the early papers that we did on TWiP just an episode ago, this raises more questions than it answers. And that's a very good sign of a paper that touched on an area that nobody has been paying attention to.
Yeah, I guess it did. Do I have something else to say? So right now there's a paper that is under review. Hopefully it'll be published, where we're addressing that issue that we'd actually discussed in a prior TWiP, where the avian malaria and certain Arctic birds will die. And at least the paper that is out there right now that I'm one of the authors on is looking at the fact that there's certain particular subspecies that are associated with clinical illness versus others that are actually well tolerated. But then it becomes interesting. I would just love to basically take the sequencing that we have and just get a bioinformatics expert to really just blast through and see, can we find any of these known viral sequences?
Interesting.
So yeah, that was...
All right. Now it's time for a hero, Dickson.
It is. And this is a hero that's suggested by Kevin. I'm not sure it's the same Kevin that we've had.
Yeah, it's the same Kevin.
It is the same Kevin. Great. So he contributes this TWiP hero for our consideration. So he says, for your consideration for TWiP hero, Rose Ghana Famben Leke, PhD. These are my notes. Dr. Despommier's stay at Notre Dame may have overlapped with Dr. Leke's undergraduate days, 1966 to 1969. In fact, that's exactly the time that I was there. But she was at St. Mary's. She wasn't at Notre Dame. She was across the street at the Women's College, which has now been, it's still in existence, and it's still an all-women's school, but Notre Dame has gone co-ed since then. So a lot of the people who would have ordinarily gone to St. Mary's have now applied to Notre Dame as well. This parasitology hero is an African woman whose principal life's work has been the study of Plasmodium and its effect on pregnant women. She has had many other scientific interests, as a survey of her biography demonstrates, with publications on onchocerciasis, trypanosomiasis, polio eradication policy, Vincent, a little touch there, epidemiology and public health and health policy. She has raised 4 children in addition to her research, so she does genetics research as well, faculty position, WHO, and various government consultancies. Dr. Leke was born in 1947 in Banso, Cameroon. She received her baccalaureate degree in 1969 at St. Mary's of the Woods, South Bend, Indiana, the sister school, as he mentioned, of Notre Dame. And she got her master's degree at the University of Illinois in Urbana in 1970 and her PhD in 1979 at the University of Montreal. She is currently Emeritus Professor of Parasitology and Immunology at the Faculty of Medicine and Biomedical Sciences, the University of Yaoundé, Cameroon. Some of Dr. Leke's recent awards and recognitions include a presidential degree in November of 2002 that appointed her the chair of the board of directors of our National Medical Research Institute in Cameroon. She, in 2011, Kwame Nkrumah Scientific Award for Women, the best female scientist in Africa. That's an enormous accomplishment. 2015, she received the title of Honorary International Fellow of the American Society of Tropical Medicine and Hygiene, a designation that formally recognizes individuals who have made eminent contributions to a particular aspect of tropical medicine and hygiene. Dr. Leke is one of 9 women to be recognized at the 2018 World Health Assembly as part of the Heroines of Health Honors. She says that her most influential mentor was Paul Silverman, and I absolutely couldn't agree more with her. Paul Silverman was the chairman of biology at Notre Dame, and he was a wonderful man that worked on tapeworms, but he had a global view of parasitism and was a wonderful mentor who supervised her master's degree at the University of Illinois in Urbana. Asked in a Lancet interview in 2006 how she relaxes, she replies, "What does that mean?" Sounds like Vincent. Explaining her early interest in the intersection of pregnancy and malaria, she says, we wanted to better understand the reasons for the disproportionate morbidity of malaria in pregnant women, how to diagnose placental malaria, and how the presence of the malarial parasite within the placenta affected the immune development of the newborns. All of these studies were conducted in partnership with my long-term collaborator, Diane Taylor, who I also know, who was initially at Georgetown University, but is now at the University of Hawaii. And Dr. Leke, despite being emeritus professor, is still very involved in research, supervising graduate students, and serving on government and international public health panels and policy-making boards. And then a lot of her accomplishments are listed in references that Kevin supplies to us as footnote. So I'm glad to recognize Dr. Leke as one of our heroes, and we would certainly get in contact with her and try to get a portrait so we can include her in our next edition of Parasitic Diseases.
Yeah, there's one on that website.
Oh, good. Good. Maybe we can use that then.
Okay. Thank you, Dickson Despommier.
You're welcome.
Now it's time for a new case study.
Absolutely. Bring it on, Daniel.
Where are we today, Daniel?
You know, I had my folder here, and I was all ready to present a case of let's see what it says here, because I'm not going to present, of a 12-year-old living in a refugee camp. But I just yesterday went to the hospital, and 2 of my gastroenterology buddies are like, Dan, you got to see this.
You got to see this. Okay.
Got to see this. And so here's the story: is there was a successful individual living in the New York area who was having several months of epigastric pain.
Months.
And so this is a man, a little bit senior to me, a little bit junior to Dickson.
That places it pretty well. Thanks, Daniel.
And when they were not sure what was going on with the epigastric pain, they decided to do an upper endoscopy, which is where they pass the scope down into the stomach, into the beginning of the small intestine. And when they got to the stomach, they saw several small, I'm going to give you a size of several small, mobile, white, serpiginous, moving objects.
Stomach?
And so I look down, and I I'm actually watching the video with them, and the first thing I want to know is you're seeing the end of the endoscope has the ability to pass out little grabbers, and the little grabbers are about 2.8 millimeters. So these are pretty small, and the object is about that size. So we're looking at a little white mobile worm-like. Little bit fat, a little chubby, and it is about 2.8 millimeters. And I ask a little bit more. Now, what they notice is a few of these are trying to burrow into the gastric mucosa.
I got it.
They're able to, so not 28 millimeters, but 2.8. So they're really small.
Sorry, yeah.
So, and they're able to grab one and send it off to the lab. So we're actually going to get a confirmation from the parasitologist of what is this thing. And then I'm going to give you a little bit of history. So this man has a curious habit of curing his own protein sources. So curing,
in quotes,
C-U-R-I-N-G, curing, which I guess he, like, if they're ill, he makes them better. No, so it's
brining or smoking or salting or something to preserve.
Exactly. Yes, he cures his own protein sources.
Right.
Well, like meat and fish and that sort of thing.
That sort of stuff. So I basically said, hey, I think it's X. And then we have our, and so I'm very curious what people think of.
Tell us how long this lasted, his symptoms.
So the symptoms have been going on for several months now.
Several months.
So this wasn't an acute, this was
several months.
Several months I've been having this issue.
Now, Daniel, does this gentleman ever go out to eat?
Yes, they certainly go out to eat.
Where did he eat several months ago?
So he eats all around. He's actually quite a fan of going out for meals.
Yes.
Yes.
Any particular cuisine that he likes?
No, pretty flexible. Why do you ask? What are you fishing for there?
I'd like to know if he's been to a sushi restaurant lately or a restaurant that's served raw fish.
Okay, so those are good questions.
But you don't know if he didn't tell you that.
I wasn't answering. I was dodging your question. I was saying those are good questions.
Okay.
See me after class.
Are there any? Does he keep nuts in his house?
No, his in-laws don't have.
No travel. If you wanted to know that, no toxic.
Does he consume his own cured meats?
Oh yes, yeah. That's why he's
routinely
curing them for his own personal consumption.
He doesn't share them with anyone?
Perhaps he does. That's a good question, too.
So,
and is it just meats, or are they meats and fish?
These are all good questions.
Okay. And no one else, does he have a wife and children?
Again, good questions.
And pictures at 11, by the way.
None of them have this illness. Well, we don't know. Again, good questions.
These are all good history to
touching all the bases, as it were.
All right.
Yeah, because we do want to better understand. I think from what I've given you, people will be able to say, "Oh, I think this is," and then you may want to go back to this gentleman and get a better sense of the source, because we may know what it is, but we also want to prevent this from continuing, right?
But of course,
several months is a long time for this.
Yeah, but not if yes and no. It depends.
I guess if you don't pull them out, they could stay there a long time.
No, but the point is that you could be curing these things routinely.
And getting in over and over again.
Exactly right.
So that'll be an interesting discussion. Is this something they acquired several months ago and have had, or has there been multiple exposures in this person? So these will be interesting topics for our emailers to address.
I would love to know when the last time he ate one of his own products was.
Interesting questions. All right, very good.
Yeah.
Thank you, Daniel.
Whoa, that's.
Let's read just a few emails here because we have some lovely, fascinating. First one is from Jimmy, who writes, "Good evening, the Three Stooges of podcast.
I resemble that remark.
I finished the TWiP marathon and working my way through TWiP. I wanted to discuss many topics from various episodes, but I will start with a recent one, 168. We discussed during 168 that bitter flavor receptors are found throughout the GI. In many Indian cultures, natural plants have been used to resolve many parasitic diseases. Growing up in India, I had these plants in various forms, and I can vouch for their efficacy. I have provided a few articles which explore these plants' extract as possible treatments for parasitic diseases. He provides 3 links to PubMed articles, and also I found a paper while preparing for my presentation. So let's take a look here at what these are. The first one is an update review on the anti-helminthic activity of the bitter gourd. Okay. That's one. And then a second one is anti-helminthic potential of 3 flowers and leaves and fruits, whose names I don't know. And here's another one. So there are plenty of articles here in PubMed. Interesting. Apoptosis mediated by leishmanicidal activity of a plant.
Right.
And the article he found when preparing his presentation, parasite-derived proteins for the treatment of allergies and autoimmune diseases, which, of course, is a different topic. Right?
Yeah.
Well, thank you, Jimmy.
But people, they make fun of that literature, but they shouldn't. Because,
I'm not making fun of it.
No. I said people, not you. But they would say, how silly of you to go out and look at, we want to synthesize some drugs.
These are called natural products. They're the sources of treatments.
So what do you think the most popular 2 drugs in the world are by far?
Quinine?
Yes. Aspirin.
Aspirin is from where?
It's from the willow tree.
The willow tree.
And the bark.
No, that's the cinchona. That's for the anti-malaria. Yeah. But the willow tree, the leaves, the Native Americans used to chew them.
Daniel, in your travels, have you ever encountered individuals who use plants to make themselves feel better?
You know, when you were reading this, it reminded me of a case I had about a year ago now, and it was a woman from India who had dengue, and her platelets were dropping precipitously, and they had dropped to less than 20. And I was actually quite worried about her. Wow. And normally, how do we approach dengue? Supportive care. But I come, and I don't know if I've mentioned this before. I come back the next day, and they've gone from 20 to 50. The next day, they're 100. The next day, they're normal. And, that is not normal. So I said to her, I'm very curious. Is there maybe something you wanted to share with me? Because you got better really quickly. And what she shared with me was that what she and other people from her region in India do is they keep aloe plants.
Right.
And what she was doing there was a certain aloe extract that one of her family members had prepared for her when they heard she had dengue. And when her platelets had gotten down to 20,000, she had started it. And then I actually went through the literature, and there were a couple of Lancet articles. So I am very humble and always willing to learn, and certain that there is so much out there that we still
sure.
So
I'll take it. Bob writes, Dear TWiP, there are now reports of a brain surgery where a tapeworm colony was removed, and he gives the reference for that. The initial reports were that the surgeons were removing a tumor from the brain when the discovery took place. Well, when you say discovery, the surgeons discovered it, but the organism that causes that is well known. And there are a series of tapeworm, larval or a juvenile tapeworm infections that you can catch from animal sources. Mostly it's zoonotic. And one of them is called Conurus, which is not a colony of organisms necessarily. When you see it, it's a sort of a large watery cyst inside of which you would find, like those Russian dolls, some stages of the parasite. Inside of that, you would find other stages that are smaller yet. And if you eat the whole thing, you get the adult tapeworms if you're the right
Tinea solium.
No. That's only one.
That's what they say she had.
Well, it's not a colony then.
Yeah, it's sort of the article's a little strange because it's not a tapeworm colony.
Well, if they said colony, then that was wrong. Because there are some that there's Tetratheridia, for instance, which sort of buds off from a common stem, but each of the buds could result in a different tapeworm. So that would be a colony. That's what I would think.
Yeah, this was actually a single neurocystic sarcosis lesion.
So neurocystic sarcosis.
And she really did need surgery for that.
It's very common.
She did not, Daniel?
Did.
No. So it's not.
Okay.
You really don't need to go in and have surgery for a neurocystic sarcosis cyst. You can either let it undergo its normal evolution. You can treat it to accelerate that, sometimes with steroids, but you really don't need to be cutting people's heads open. So, I know when I looked at this article and, she's happy, surgeon's happy, I was kind of not so happy.
Surgeon's very happy.
But yeah, you really, this is the number one cause of seizures throughout the world.
Right.
And, this happens and you really don't need people to have their heads cut open.
Well, I would, I'm a non-physician, so I can't really disagree with what you're saying, but I would like to add something to that, and that is that there are some circumstances when you do the CAT scans and you find out where this lesion is, that it's right at the surface of the brain, right? And a lot of surgeons are tempted to just go in and take it and get it out of there, right? Because they know that it shouldn't be there. But most of the lesions are in the deep brain tissue, where if you go in to get this cysticerca stage, you're going to mess up all kinds of neural networks that the patient may never recover from again. So they did some studies on whether you should or shouldn't operate, and whether you should. I think the treatments now have gotten better with drugs, so that you can treat with high doses of, I believe, they're using metronidazole and several other drugs in order to interfere with the biology of this parasite without destroying the brain tissue. So, but not an unusual finding. Unfortunately, it's not an unusual finding. But thank you for pointing it out to us.
So it happened at Mount Sinai here in New York.
Yeah.
Daniel, can you read Ben's? Sorry.
Ben writes, "Dear hosts of podcasts on parasites, I would love it if you could share this on the TWiP. There is a parasitology course run by the Australian Society for Parasitology called Concepts in Parasitology, running in November this year. I took the course 2 years ago, and it was absolutely fantastic, covering everything from tongue-snatching parasites of fish to electron microscopy of toxoplasma. It is also where I took the Toxocara canis image I sent in a few weeks ago. Down under is a long way to travel for those outside of Australia, but I promise it's worth it. You'll stay on the beautiful east coast of Australia and be greeted every morning by more kangaroos than you could possibly dream of. Check it out." And he gives us a link, which I guess will be in our show notes.
Right. Dickson, you going to go?
I've already been there. I spent 6 months in Australia on my sabbatical. I wish I could go actually back. I would like to go back and revisit the Walter and Eliza Hall, for instance, where I had some really good months.
The next one is from Octavio. Octavio is a veterinarian from Portugal, and this is a late response to the
case.
The case of a 10-year-old boy with, I guess he did have cutaneous leishmaniasis, right?
Yes.
But I wanted to point out that he says, being a veterinarian, it's not correct for me to suggest a treatment. But if this was found on a dog, meglumine antimoniate or glucantime, allopurinol, and miltefosine would be the drugs of choice, given in different protocols depending on the International Renal Interest Society staging of the animal's kidney disease. The most common systemic consequence of the disease, and that is the main driver to the fatal outcome in dogs. And then he says, I would like to include in this email a human case which happened in Portugal. And
I guess
he doesn't want us to disclose it in case we ever want to use it.
All right. So he should, Octavio, send us the story. We'll look it over, and if it's
he did. It's the link here.
Okay.
So we'll take a look at it, Daniel, and decide if you want to present it.
Oh, that looks great.
Sometime.
All right, I'll check it out.
It uses dermoscopy, which you mentioned before, Daniel.
That is perfect.
All right, let me take Jenny's. This is a technical thing. Jenny writes, "Hello there. I love your podcast, and I am also watching the YouTube videos. I have a Kobo. I tried to look for your book on the Kobo store, but could not locate it. What other places could I download it from?" So, Daniel, I didn't know this, but Kobo is another store where you can buy digital books.
Okay.
And so I know, Jenny, that you can get PD6 and 7, just 6, or is it 7 as well, Daniel, on the Amazon Kindle, right?
So you can get 7. Yeah.
7 as well. So you can get 7 on the Kindle, but you have to pay for that, right, Daniel?
I think it's like $1 or $2. It's very inexpensive.
Yeah.
And
or you could download the PDF, which I think you could put on a Kindle, right?
And the PDF's free, and you can read it on a Kindle.
Yeah. So there you go. We don't have Kobo, but you can download the free PDFs at the parasiteswithoutborders.com. There's a page now.
Right.
What did I name that page? Let me look at it.
Resources or something?
It's called Books.
Books. Okay.
Oh, books.
Called Books.
Great.
Because that's where and I thought, yes, Books, and it has PD7 and where you can get it from. It's got PD7. It's got Paul Has Measles. And the newest edition is Medical Handbook for Limited Resource Settings 6, which is a free PDF.
May I request that you might want to put up My People, Parasites, and
Please do.
You mean a link to buy it, right?
Yeah, not necessarily just put it up there to let them know that it exists.
Yeah, I put up Peter Hotez's Vaccines Did Not Cause Rachel's Autism, put up Principles of Virology, and I will put up
People, Parasites.
And Plowshares. You want me to put Vertical Farm up too?
No.
You got any other parasite books?
Well, 7th edition.
Well, that's there already, dude.
Well, you asked me.
I know. I'm just giving you a hard time.
Yeah, that's okay.
Dickson, can you take Anthony?
Anthony writes, Climate Change and Vectors. Your colleague, Jeff Schumann, is a meteorologist. I don't know if he focuses on vectors. At a glance, here, and then he gives a reference as an internet reference, shows longitudinal expansion. Looking at altitude/latitude, going up with the thermometer is a great place to start. There'll still be forces in place unrelated to climate change affecting population dynamics. And even with climate change, there'll quickly be a who's on first, what's on second of changes in hosts, for example, bird migration patterns, humidity, vegetation, pathogens/predators/parasites of the vectors, etc. These will be the ingredients of a stew of a complex model.
So the paper he sent is in Journal of Medical Entomology. It's called County Scale Distribution of Ixodes scapularis and Ixodes pacificus in the continental U.S.
Wow.
These are agents of Lyme disease, right?
Yep. And of other things,
carriers,
too.
vectors. And they're talking about the expansion of the range. Majority of county status changes occurred in the north central and northeastern states.
Northeastern, yeah.
The south remained fairly stable.
Yep. I would agree with that.
All right. And the last one, Daniel.
And writes, hello, TWiP professors and doctor. Another article extolling the virtue of ivermectin, which I've heard mentioned often on your podcast. And she gives a link, and it's an article in the New York Times about scabies treatment with ivermectin, which I think we mentioned earlier on the show.
We did. It's a
really?
Nice double hit.
Yes. Remember we said instead of covering that guy with like shampoo and permethrin, we could just give him ivermectin.
Yeah.
So scabies means misery. This pill can end it.
Exactly.
Nice.
And they've got a great EM colorized scabies.
Pretty scary.
Yeah, actually that is.
All right. That is TWiP 173. A little bit longer, but we haven't done it for a while. So there you have it. You can find it at microbe.tv/TWiP. And you can also find it if you listen, of course, on a podcast player, which is on your phone or tablet. Mine of choice on my phone is called Overcast. Isn't that cute? Podcast, Overcast, yep. Overcast skies, you know.
Indeed.
Please subscribe if you listen to a podcast player. Why? Because that way you get every episode as we release it automatically. It's free. And we know how many people are listening. That's very important to us as we move forward to make our podcasts more sustainable. I don't know if you can be more or less sustainable, Dickson, but you're either sustainable or not, right?
I'll let you know.
It's a binary thing.
It certainly is.
So please subscribe. And if you very much enjoy what we do, as Volker asked at the top of the show, please consider giving us $1 a month. You can go to microbe.tv/contribute. You could use Patreon. You can use PayPal. It would really help us out a lot. Thank you.
Do you realize if you had $1 for every person alive today on the planet, you'd be very wealthy?
Well, I'd be a 7 billionaire.
You would.
And I would only need it once.
$1, that's all it would take.
Well, I don't need $1 from everyone. I would love $1 from everyone listening every month.
Every other one.
It's $12 a year. It's not a lot.
No, it isn't.
And with that, we could do great things.
We can do more than we're doing now. That's true.
So please help us out, microbe.tv/contribute. And of course, send your questions and comments, your guesses for this episode's clinical case. 3 months of epigastric pain.
With little white things.
Was it 9 months?
9 months.
Let me make sure I put that there. This is several, so I'll put 9 months. That's a long time.
That is a long time.
It's like being pregnant and suffering the entire time, right?
Indeed.
TWiP at microbe.tv. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Always a pleasure.
Your redness has subsided.
Yes, yes.
Daniel went running just before.
He did.
And it's 90 degrees out, right?
It is hot.
He was hypoxic when he came in.
Celsius.
That's right.
Dickson Despommier is at tricanella.org and thelivingriver.org. Thank you, Dickson.
This was wonderful return to routine that I truly enjoy.
Dickson, we just did 2 podcasts in a row consecutively.
We did.
From 2 to 6 p.m. we've been podcasting.
Because we love to.
I do. I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiP is by Ronald Jenkins. Thanks to ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.